So I'm Professor Karen Esler. I am a professor at Stellenbosch University and I work in the Department of Conservation Ecology and Entomology. And a lot of my students are really at the interface between application and research. So they're working in used environments and environments where we need to understand management actions. The challenges and practical solutions that we've seen unpacked in this field trip provide a window into the complexities that may be experienced in riparian restoration or rehabilitation projects elsewhere. And as we scale up our efforts in the United Nations Decade on Ecological Restoration, our role as researchers is to ensure that the lessons that are learnt on the ground can be replicated and can provide an objective evidence base so that we can put effective policies and management strategies in place elsewhere. So in South Africa, we have a relatively small and well-connected ecological and environmental management community, making it really possible for us to engage and collaborate across what we call the knowing-doing gap or the divide. And the Berg and Breede rehabilitation program is really fantastic and a really great example of this, because it's enabled us to consider the complexities of that emerge when we scale up our rehabilitation activities. And at the Department of Conservation Ecology and Entomology at Stellenbosch University, our focus is on biodiversity conservation, and particularly the focus is on used environments. And so the partnerships that we have built with practitioners on the ground, like in Thaba, and specifically within the Berg and Breede catchment, have enabled us to move beyond experimental plot-based research and to focus on a landscape level, which is really amazing. It has allowed us to focus on multiple sites across the landscape. And we have seen that through doing that, we can really understand some of the complexities that emerge from moving beyond or scaling up. And I think that's actually quite an exciting partnership to have. It also allows us to decide where and when, for example, we should be applying active restoration measures into the landscape. So there's quite a few really cool bits of research that can go on in the Berg and Breede catchment in association with those partnerships.
The following 3 inserts will give more insight into some of the research conducted on the Berg and Breede project. Dr. Charlene Janjan-Skeepers is a terrestrial invertebrate ecologist and physiologist. She mainly works on soil systems that are key for terrestrial ecosystem functioning, using springtails, Collembola, as exemplars and adopting a multidisciplinary approach incorporating physiology, traditional taxonomy, DNA barcoding, and ecology. Monique Van Zitters is currently researching active rehabilitation methods on rehab success and key variables to success in the Berg and Breede catchment areas. Georgina van Biljon is researching success factors in the riparian restoration, focusing on plant survivability on both the rehab sites as well as in the nursery.
Invertebrates make up 95% of the living planet. Despite this, we don't know much about them, especially their role in the restoration process. We work on Collembola or springtails. They are tiny little invertebrates that live in the soil or the leaf litter, and they're extremely abundant. Springtails are an ancient group of hexapods, which means they have 6 legs, but they do not formally form part of the insect group. What makes them very different to insects is that they do not have wings, they're wingless, and they have a special feature called a furca, which is an appendage underneath their bodies, which is used to escape from predators. So when predators come close to them, they release their furca, and then they can escape by jumping. They also do not have an exoskeleton like many insects. So insects have an exoskeleton, which protects them from desiccation. Collembola does not have an exoskeleton, which means they're very prone to desiccation, but it also means they're in direct contact with their environment. And this makes them really excellent indicators for ecosystem health or environmental health. Globally, there are about 8,000 species in the world. In South Africa, we know of about 150 species, although we expect hundreds more to be discovered and described. Calimbula are very important decomposers in the soil ecosystems. And by this, I mean that they are breaking down leaf litter and thereby indirectly producing or supplying nutrients to the plants. They are also very, very important prey to predators such as spiders, pseudoscorpions, and predatory mites. So having a healthy ecosystem, having a lot of Calimbula in the system, means there are lots of predators, which in turn means pests can be controlled without the use of pesticides. We are looking at how the soil fauna is recovering after the removal of invasive alien plants. And we're finding quite interesting results if we compare the passive and the active restoration sites. Firstly, we're seeing that the active and passive restoration sites are not really that different in terms of the number of springtail species, also the number of species, or the abundance. So what we're finding, though, is that it differs in the community. The species that occur in the passive site is not necessarily the same species that occur in the active site. The below-ground fauna takes much longer to recover during a restoration process than the above-ground.
My name is Monique Van Zitters, and part of my research is evaluating the effect of active rehabilitation measures on rehab success and key variables that affect success in the Berg and Breede catchments. So we're basically investigating the effect of active rehabilitation on the native vegetation recovery, and as well as looking at the costs or the effect of investment on the rehabilitation success. And this was conducted in 11 sites across the Berg and Breede catchments. So some of our main findings were that both active and passive rehabilitation sites showed native vegetation recovery, rehabilitation success, but active measures resulted in a slightly enhanced recovery. Native species cover, richness, diversity, and evenness was slightly higher than the active sites. We found that soil and geomorphological variables were the same across sites, and that suggests that the rehabilitation success was due to the method, the actual rehabilitation method, and not the abiotic conditions. A sufficient monitoring period needs to account for lag effects, especially for native vegetation recovery and abiotic conditions.
Intaba is currently recording success factors in the nursery and in the field. In the field, we are undertaking 3 different forms of research. We're looking at the plant survivability on 4 sites in the Berg catchment. The 4 sites are within a 10-kilometer radius, and we are looking at monitoring 22 plant species. On the sites, we plant on dripper lines, and we have selected a sample of the dripper lines and record the plant height and survival on these sites. We're doing fixed point photography on all of our sites, and we're also looking at changes in plant community and structure over using fixed line transects on 12 sites over a 3-year period. And we're also doing pilot studies on the effect of biochar on plant growth and survivability. And this is, we are looking at 10 different plant species. And we also are doing research in our nursery and looking at cutting success rate, seed germination, and transplanting success. The results will be available in March 2022. Our funding cycles are over a 3-year time period. In the last 6 years, we have undertaken an average of 4 rehab sites per year. In the next 3 years, we will be doing between 20 and 40 rehab sites per year with the help of our partners to undertake the rehab. Our rehab sites are between 3,000 to 5,000 meters squared, which is an average of 2,000 to 5,000 plants planted per site.
Intaba relies greatly on our staff to assist with the research in the nursery and in the field, namely with measurement and data capture. We also learn a lot in partnership with others by looking at things through another set of eyes, making us stronger through diversity. We are observing many interesting things in our field, from the recruitment of small wild olive trees under a tree on a 6-year-old rehab site, to what germination strategies work for specific species. There are still many stories to be told and to be investigated.